Hello everyone. In this session, we are going to discuss a small yet very important topic, that is microscopes. You frequently get question in your exam on the microscopes, MCQs also, and in university exam also usually you get shorter, very short question, right? Now we know microbiology is nothing without microscopes. We can't see the tiny organisms, that is bacteria, virus, fungus or parasite, without microscope. So it's very important to understand the microscopes to understand the microbiology. So let me share my screen quickly with you. I guess you can see my screen, and let me start this topic, the microscopes. So I welcome you all for this Profbuster Live Progenitor batch, and here today's session is on general microbiology, and the topic of general microbiology is microscopes. This is my overview under which I'm going to discuss the topic microscopes. If rarely you are getting a long question on the microscope, you have to describe under these heading, but usually you get short question on one of the type of the microscope. So first of all, I will let you know the introduction, then I will let you know the 3 important properties of the microscopes, and then I will tell you the 5 type of the microscopes. What 5 type of microscopes? Bright field microscope known as light microscope, dark field microscope. Okay, this is bright field microscope. This is dark field microscope, phase contrast microscope, fluorescent microscope, and electron microscope. There are 5 type of microscope. You should know the principle of each of them and use of each of them, so that we will make a master table, a master chart in which we will discuss all 5 type of microscopes. Okay, in the end, I will let you know the differences between light microscope and electron microscope. The light microscope is the first one, the electron is the last one. You should know the differences between them. So these are the questions which are frequently asked from this session, and we are going to continue in this sequence. So let's start it. Let's start with the introduction. Okay, in the introduction, I would like to tell you the definition of microscope. What is microscope? What do you mean by microscope? Microscope is a special instrument. It's a specialized instrument which is used to view the objects which cannot be seen with naked eye. With naked eye, we are not seeing any organism. We are unable to see because that object is very small. It is not necessarily an organism. It can be any object. It is very small. So we need to magnify it. And for magnifying, we need a special instrument with a magnifying glass and that is known as microscope. So you should be able to define it. What is microscope? It's a specialized instrument which is used to see the objects which cannot be seen with naked eye. Now, to understand this, you have to understand a term. The name of the term is resolution power. What do you mean by resolution power? First, you tell me what do you mean by resolution power. Resolution power is the ability of human eye to see 2 separate images of closely placed objects. Okay, let me ask you a question. Can you see 2 dots separately? Yes. Can you see 2 dots separately? Yes. Can you see 2 dots separately? Yes. Can you see 2 dots separately? Still yes. Can you see 2 dots separately? You will say no, ma'am. Even I can't see them separately, but they are 2 different dots. If we use a microscope over here, we will be able to see them separately. You are getting my point? So the ability of human eye to see 2 separate images of closely packed objects. These are 2 closely placed objects. These are 2 closely placed objects. Now I want to see them separately. I want to see 2 separate images, like here 2 separate images, 2 separate image, 2 separate image, 2 separate image. But here they merged. Here I didn't got 2 separate image, I got 1 image. Okay, so I want 2 separate images of that. That is known as resolution power. Resolution power. So how much minimum distance should be there between the 2 objects so I can see them separately? Okay, below that distance, they will merge with each other. So you should know the minimum distance for the naked eye, the meaning of the resolution power. Okay, here you can see the 2 images separately. Here you can see the 2 images little bit separately, but here they completely merge. Here they started merging and here they completely merge. Okay, so what is resolution power? The resolution power is the ability to see 2 separate images of closely placed object so that we can distinguish them as 2 different entities. Okay, here I can say it is 1 entity, but actually they are 2 entities which merged with each other. That is the meaning of resolution power. So what is the resolution power of human eye? Human eye, okay, naked eye, human eye, unaided human eye. We are not using any magnifying glass. If I use the magnifying glass, the resolution power will increase, but I am not using any magnifying glass, so unaided human eye, it is 200 micrometer. So the distance between 2 points should be 200 micrometer if you want to see them separately. If the distance is less than 200 micrometer, they will merge. So minimum distance between 2 objects for unaided human eye is 200 micrometer. But the point is that bacteria are smaller than that. Bacteria, virus, fungus, parasite, they all are smaller than that. So we can't see them with naked eye. We need a microscope. So you should be able to explain why we need microscope, because the resolution power of human eye is 200 micrometer, or in terms of meter, you can millimeter, you can say 0.2 millimeter, you know, 0.2 millimeter. So either you say 200 micrometer, micrometer is 10 power minus 6 and millimeter is 10 power minus 3. You can see we are doing the conversions. Okay, so either you say 200 micrometer or you say 0.2 millimeter, whatever you say, the meaning is the same. So distance between 2 points, minimum distance, these are 2 points. If I want to see them separately, let me erase these annotations. Yes, so these are two different points. If I want to see them separately, the minimum distance between them should be 200 micrometer or 0.2 millimeter. The meaning is same, the units are different but meaning is same. If the distance is less than that, okay, the 2 points will merge with each other and we will see one image, not two different images. So this is the resolution power of human eye. The resolution power of human eye, please learn it. Learn resolution power of human eye. The resolution power of human eye. What is the resolution power of human eye? Say 200 micrometer or 0.2 millimeter. This is resolution power of human unaided eye. Okay, but the point is that what is the problem? The problem is that microorganisms, bacteria, virus, fungus, and parasite, they all are very small. Okay, they all are in micrometers only, less than 200. So we cannot see them separately. And viruses are even in nanometer. Virus to nanometer mein hote hain. Okay, the bacteria, fungus, and parasite are in micrometer, 10 power minus 6. But viruses are in nanometer, 10 power minus 9. Okay, so they are not within resolution power of human eye. So with naked human eye, we cannot see them. We cannot see them. So we need a special instrument that will magnify them. Okay, that will increase the resolution power, and that is microscope. So we need a light microscope to see the bacteria, fungus, and parasite, and we need an electron microscope to see the viruses, because viruses are in nanometer. They are very small. Bacteria, fungus, and parasite are small, but they are in micrometer. Okay, but viruses are too tiny, 1,000 times more tiny as compared to bacteria, fungus, and parasite. So we need a higher grade microscope, a more magnifying microscope to see them. So we see bacteria, fungus, and parasite with light microscope, but viruses with electron microscope. I hope you got the concept. Okay, you can see bacteria, fungus, and parasite. They are in micrometer. Okay, so we need a light microscope to see them, but viruses are in nanometer. So we need an electron microscope to see them. That is the point, because they all are below the resolution power of human eye, that is 200 micrometer. They all are below. So with human eye, with unaided human eye, we cannot see them. We need microscope. So that was the introduction. In the introduction, I taught you the definition of microscope. What is microscope? I taught you the definition of resolution power. What is resolution power? Okay, I told you what is microscope, what is resolution power, and I taught you how we can increase the resolution power, and to see bacteria, virus, fungus, parasite, we need microscope. So that was the introduction. Now coming on the properties of microscope. Properties of a microscope. There are 3 properties of a good microscope. Okay, there are 3 properties of a good microscope. The microscope should have good resolution, good contrast, and good magnification. 3 properties: good resolution, good contrast, good magnification. Let me explain you. Resolution, I already taught you. What do you mean by good resolution? What is resolution power? Resolution power is the ability to produce 2 separate images. It's the ability to produce 2 separate images of 2 separate objects. They are not merging with each other. Okay, so what should be the minimum distance between them? Okay, so that is the resolution power. How to increase the resolution power? How to increase? We can increase. Now there is a principle in physics. Resolution power is directly proportional to refractive index of the medium. You know, refractive index of the medium is known as mu. In physics, you may have studied it now. So resolution power is directly proportional to mu. If you increase the mu, resolution power will increase. What is mu? Mu of the medium. Okay, you can see this is our microscope and this is our slide. Our slide on the stage of the microscope. Can you see the slide is placed? So on the slide, we have the bacteria, virus, fungus, parasite, whatever. And this is the lens. So between the lens and between the microorganism, we are having air in between. Normally, we are having air in between. So air have a mu. Okay, but the mu of oil is more than that of air. So what if the medium between the object and the lens is oil? So that's why we put 1 drop of oil. You must have used oil immersion lens, na? So we put 1 drop of oil on the slide and we put lens in that. So the medium air we are replacing with the oil. So we are increasing the mu. So we are increasing the resolution power. I hope you got it. So the mu of oil is more than mu of air. And that is how we are increasing the refractive index by use of oil in the oil immersion lens. Okay, you can see how we increase the resolution power. You can see here. Can you see the points separately? Yes. Here they are little bit merging. Here they are completely merging. Here they are completely merging. You are getting my point, huh? So for unaided human eye, the minimum distance between 2 points should be 200 micrometer. Then only we will be able to see them separately. But if you are using light microscope, so we have increased the magnification by 1000 times. So the minimum distance between the 2 points is not 200 micrometer. Instead of 200 micrometer, it's 0.2 micrometer. Just 0.2 micrometer. If the 2 points are just 0.2 micrometer away, we will be able to see them separately. But in electron microscope, it is not 0.2, it is 0.5 micrometer. Okay, it's not 0.2, it's 0.5 nanometer. Please do correction. It is not micrometer, it's nanometer. It's nanometer. Okay, so even the distance is very less. Okay, so for human eye, for light microscope, for electron microscope, tell me the resolution power. Tell me. So what should be the distance between 2 points so that we can see them separately? What should be the distance between 2 points so we can see them separately? What should be the distance between 2 points? So here the distance between 2 points should be 200 micrometer. Here the distance between the 2 points is 0.2 micrometer. And here the distance between 2 points is 0.5 nanometer. Okay, even the 2 points are just 0.5 nanometer away. We can see them separately. So highest magnification is seen in electron microscope. And we can increase the resolution power by using oil immersion lens via increasing the refractive index of the medium. If we are increasing the refractive index of the medium, we are increasing the mu. We can increase the resolution power. Resolution power is directly proportional to mu of the medium. That is refractive index of the medium. That is a principle in physics. And we have applied that principle. So I hope you got it. Okay, I hope you got it. Now coming on the second property. The second property should be good contrast. The microscope should have a good resolution. So you can see this is not a very good microscope. This is the average microscope, but this is the best microscope because this microscope can separate all the points separately. Okay, here the resolution power is very high. We can increase the resolution power by using the oil as a medium between the object and the lens. Okay, via putting 1 drop of oil. Okay, now I want to increase the contrast. The second property of a good microscope is good contrast. How we can provide contrast? You know what's the problem? The problem is that slide is transparent. Everyone knows slide is transparent, but how many of you know bacteria is also transparent? Yes. So can you see a transparent object on a transparent background? Suppose I'm putting a bacteria over here. Can I see the bacteria directly? No, because this is transparent, this is transparent. Both are transparent, there is no contrast. For seeing anything, human eye need contrast. We need contrast. The colors should be different. My color is different. My background color is different. Suppose the background is also black. I am completely black. So you cannot see me. So same object and same background you cannot see. The background and the object should have different color and that is known as contrast. Here the object is transparent. Okay, the slide is transparent. Both are transparent. You cannot see them. Okay, you need contrast to see anything. That's why we will stain the bacteria so that the color of the bacteria will be different as compared to background, and that is staining. So if you want a good contrast, stain your slide before putting in microscope. If you are directly putting the slide in microscope without staining the bacteria, you will not see that. You will not have good contrast. So first you stain it. You can stain using Gram stain, Zn stain, Albert stain, Jimsa stain. Many stains are available. Use any stain. You stain your object and then see under microscope. You will get a good contrast. See this image. In this image, we have used a blue stain. So all the bacteria become blue, but the background is still transparent. Appreciate the background. The background is still transparent. The bacteria are blue. If we don't stain the bacteria with blue, the bacteria are also transparent. Imagine bacteria is transparent, background is transparent, we cannot see them. So to provide contrast, we need to stain the specimen. And finally, the third property. The third property is good magnification. The magnification should be good. Okay, so good resolution. Okay, good resolution power, good contrast, and good magnification. How to increase the magnification? We have to magnify the object. No, we have to magnify it. How to magnify? So in a light microscope, we are having 4 types of lens. First of all, we are having a scanner. The scanner is 4x. Okay, we are having 2 lenses first. You can see this where we put the object, where we put the slide. We put the slide over here on the stage. This is stage on which we put the slide. So this lens are known as objective lens. These lenses, which are of 4 types. Okay, and here we put our eye. Okay, we put our eye over here. Okay, and we see the object. So this lens is known as ocular lens, which is only 1 type. Ocular lens is only 10x. It will magnify the object. Whatever object you are putting here on the slide, that will get magnified 10 times by the ocular lens. But here we have 4 options what you are putting. We have a scanner that is 4x. We have a low power that is 10x. We have a high power that is 40x, and we have an oil immersion lens that is 100x. I guess everyone have used the light microscope in your laboratory. I guess you know these basics, right? So here you have 4 options. You can select it, but here you don't have any option. Eyepiece is always fixed. Eyepiece, you don't have any option. You have seen the microscope, light microscope present in every laboratory in medical colleges. I guess you have seen that. So ocular lens is 10x. But objective lens, where we are putting the object, okay, first you have to see on the scanner, so it will be 4 times magnified. Okay, then you have to see on low power, it is 10 times magnified. Then you have to see on high power, it is 40 times magnified. And if you are putting a drop of oil here and you are using oil immersion, it is 100 times magnified. So this is how we use the magnification. Ocular lens is always 10x. Always 10x. There is no option. But objective lens, here we are having 4 options. 4x, 10x, 40x and 100x. Here 4 options. So final magnification will be what? Suppose whatever is my object is 1 millimeter. Okay, 1 micrometer. So how many times it will be magnified finally if I see them over here with the eye? This is my eye. How many times it will be magnified? So here it is 10x. And here, whatever you are choosing, you have to multiply that. If you are using 4x, so 4 into 10. If you are using 10x, so 10 into 10. If you are using 40x, so 40 into 10. If you are using 100, so 100 into 10. Suppose you are using 100. So 100 into 10 will be 1000. So whatever object you are using, it will be 1000 times magnified. So total magnification is the multiplication. See the multiply sign. It is the multiplication. Multiplication of the magnifying power of the ocular lens, which is always 10. And the magnifying power of the objective lens, here we are having 4 options. It can be 4, it can be 10, it can be 40, it can be 100. So we have to multiply 10 with either 4, either 10, either 40 or either 100. This is final magnification. This is how we calculate the final magnification. So what are the properties of a good microscope? There are 3 properties of a good microscope. What are the 3 properties? Good resolution, good contrast, good magnification. How we can increase resolution? What is the summary? How we can increase resolution? What is resolution? Ability to see 2 separate images of closely placed objects. Okay, so we can increase resolution by putting oil as a medium, because the mu of oil is more. Mu of oil is more as compared to mu of air. The refractive index of oil is more as compared to refractive index of air, and resolution power is directly proportional to mu. So if we are increasing the mu, the resolution power will automatically increase. That is the principle. You have to explain it. Number 2, how to provide a good contrast. Stain your specimen, stain your bacteria, because bacteria are transparent. And how to increase the magnification? By applying convex lens at the ocular lens as well as objective lens. Ocular lens, we have only one option, that is 10x. But objective lens, we have 4 options: 4x, 10x, 40x, 100x. The final magnification is the product of the two. So 10x is multiplied with either 4x or 10x or 40x or 100x. So this is how we calculate it. So the 3 properties of a good microscope is good resolution, good contrast, and good magnification. All the 3 properties should be present in a microscope, then it is a good microscope. It should be able to increase the resolution power, it should be able to provide a good contrast, and it should be able to provide good magnification. Okay, so it's a good microscope. So you frequently get question on the properties or one of the property. So we have seen the introduction. In the introduction, we have seen the definition of microscope, why we need a microscope. Then we have seen the 3 properties of microscope: resolution power, contrast, and magnification. And we have seen how to increase them. How to increase resolution power? By providing oil as a medium. How to increase contrast? By staining the specimen. And how to increase the magnification? By using convex lens at the ocular lens and the objective lens and multiplying them finally. Okay, now finally we will come on the 5 type of microscope. What are the 5 type of microscope? Bright field, also known as light field microscope. Bright field or light field. Second is dark field. Third is phase contrast. Fourth is fluorescent. And 5th is electron microscope. So I want all of you to learn first 5 names. Okay, these 2 are contrast. You can see bright field and dark field. Bright field or light field is same. Bright field, light field is same. Bright light. Okay, dark field. Third is phase contrast, then fluorescent, then electron microscope. For each microscope, you should know the introduction, their definition, their principle, ultra important. You frequently get question on the principle of the 5 microscope. Okay, I will tell you a diagram. You should be able to draw that diagram like the physics, okay, and application. So this is my master chart, the master chart of the microscopes. So we will see the master chart of microscopes over here. So let's start the 5 type of microscope with their master chart. Okay, let's start with the first one. Okay, everyone with me, take out your notebook, take out your pen. I will not say again and again. Come on. So write it. Make this master chart so that you can revise it before you go to your exam. So start with the first one. The first one is the bright field or light field microscope. The first is the bright field or light field microscope. What is the introduction? Why it is known as bright field? This microscope that you use in your laboratory day to day. Everyone have used it in your pathology laboratory or microbiology laboratory. This microscope is light microscope or bright microscope. We call it bright field or light microscope. Everyone have seen that. Okay. You should be able to label this image. Sometime in the MCQs, labeling is asked. Okay, this is ocular lens, I already taught you. This is objective lens, I already taught you. This is stage, this is condenser, you can see. This is light bulb. This is light bulb from which light is emitted. I guess everyone knows the basics. Okay, now what is light microscope? Why it is known as light? Because it forms a dark image on a light background. Here the background is light, but the image is dark. I will show you. The image is dark, but the background is bright. That's why it is known as bright microscope. Based on the background, we are naming it. The background is bright. That's why it is known as bright or light microscope, but the image is dark. I will show you. What is the principle? What is the principle? This is the principle. You have to draw this diagram. You have to draw this diagram. I will explain the principle, the same diagram in terms of flow chart. So in a microscope, where is the bulb? You can see here is the bulb. Where is the bulb? Can you appreciate the bulb over here? Yes, you can appreciate. This is the light bulb, the source of light from which the light is coming. So when the light is coming and light first go in the condenser. You know the labeling now, people here. Let me show you the labeling. You can understand the principle if you can understand the basic diagram. First you see the basic diagram to understand the principle over here. So this is the light source. Okay, this is the condenser. This is the object. This is the stage on which we will keep our slide. This is objective lens and this is ocular lens. So when the ray of light will come from the bulb, first it will go in the condenser. Okay, first it will go in the condenser. Then it will go in our object. In our object, that is our slide present on the stage. Then it will go in the two lens, ocular lens and objective lens and then ocular lens. So first it will go in the condenser. Then it will go in our specimen, the slide, which contain our specimen, maybe bacteria, maybe virus, maybe fungus, maybe parasite. Then it will go in objective lens and then it will go in ocular lens and finally the image is formed. Okay, so this is the principle. You can understand it here. So first a light source is coming from the bulb. Appreciate the bulb over here. Appreciate from which light is coming. You have to draw the bulb and you have to draw the rays which are coming. They will go through condenser. So you can appreciate this is the condenser. I have shown you the diagram now. So first of all, there is the source of light. Then there is the condenser. So the condenser is hollow condenser. It's hollow. It is having only periphery and condenser is hollow. So all the light get condensed from that condenser. All the light is passing. Whatever rays you are taking it from here, all the rays, you have taken 3 rays now, 1, 2, 3. They all will pass from this condenser and they will fall on the stage. This is the stage. This is a slide. And on the slide, this is my bacteria. Can you appreciate? This is my bacteria. So the light will pass from the condenser. Then it will go on the stage, on the specimen. Can you see it here? Can you compare it? So first the light is coming from the condenser. Where is from the bulb? Where is bulb? Here is bulb. After that it is going in the condenser. After that it is going on the stage, on the slide. The slide contains the specimen. So it is going in the specimen. You can see it is going in the specimen. Okay, now from the specimen, 2 type of rays are there. See this diagram closely. You should be able to draw that. You can see here I have taken 4 rays, not 3. Total 4 rays. Can you appreciate 4 rays? 1, 2, 3, 4. All 4 passed from the condenser. 1, 2, 3, 4. All 4 passed from the condenser. Now from the bacteria, 2 of them are passed, but 2 are not passed. The one which are passed from the bacteria, they are deviated. Okay, they are reflected, sorry. And the one which are not passed, they are non-reflected. So we have 2 type of rays, reflected and unreflected. Okay, reflected, unreflected, both of them are going to the objective lens and then ocular lens. Objective lens will magnify it based it is 4x or 10x or 40x or 100x. We are having 4 options. Ocular lens always 10x. Okay, so it will magnify it. Okay, all the 4 rays are passing from here. All the 4 rays are passing from here. Ultimately, the image is formed on the eye. So it will form from the objective lens. Objective lens will magnify it. Then it will go on the ocular lens. Ocular lens will magnify it. And finally, a dark image is formed on a bright background. I hope you got the meaning of the dark image. Okay, the reflected rays are there. You can see here the reflected rays and unreflected rays both are passing. The reflected rays will give a dark image and the unreflected rays will give a bright background. I hope you got the principle. So say the principle. say the principle. You don't have to go in detail in physics. You should be able to understand it broadly. You don't have to go in each step, how it is happening. It's not a physics, it's microbiology. Okay, so you can see there is a light source. From the light source, the light is coming. The light rays, you have to draw 4 rays that is going in condenser. The condenser is hollow. Appreciate the condenser, which is hollow, from which all 4 rays are passing. And after that, it is going on the specimen. You can see this is a slide on which the specimen is a bacteria. Now from the bacteria, you can appreciate 2 rays are passing. You can appreciate this one is passing, this one is passing, but these 2 are not passing. So reflected and unreflected rays, both reflected and unreflected rays will go to the objective lens and ocular lens, and finally an image is formed. They all will go to the objective lens and then ocular lens, and finally a magnified image is formed. So the magnified image is having a dark object and a bright background. You can see the reflected rays here form a dark object, and the unreflected rays will form a bright background. That is the principle. Broadly you have to understand, and you should be able to draw it. You should be able to write it. This is the principle of light microscope. It is the most common microscope used in labs. In patho labs, in micro labs, you all have used it. So that is the principle. Okay, so in the introduction, we will write here, it will form a dark image on bright background. That's why it is known as bright field microscope. Principle, I have told you. Application, it is the most common microscope we use, and I told you. Now coming on dark field microscope. The second microscope is dark field. Here it is forming a bright image on a dark background. Okay, the name is according to the background. Why it is called as dark field? Because the background is dark. Here the background is dark. The image is bright. It's reverse of that. The bright field was forming a dark image on a bright background. This one is forming a bright image on dark background. So appreciate the background. Here the background is bright, here the background is dark. That's why this is known as bright field. That's why this is known as dark field. This is how you have to learn. So it is forming a bright image on a dark background, and that's why it is known as dark field microscope. Dark microscope or dark field microscope. Based on the background, we name it. What is the principle? Only one difference. The condenser is different here. Okay. You can see here also we have a light source. Appreciate here also we have a light source. Appreciate the light source which is emitting light. Okay. From the light source, the rays are coming. Again, 4 rays are coming. You can appreciate. Okay. But the condenser, in light microscope, the condenser was completely hollow. But here the condenser periphery is hollow, but the center is opaque. The condenser is like this. Appreciate. The center is opaque. Okay. And the periphery is hollow. You can appreciate the periphery. This is a condenser. Okay. You can appreciate the condenser is like that. It is not completely hollow in contrast to light microscope. In dark microscope, the condenser is a special condenser. Here is a special condenser which is having a central opaque area and periphery hollow area. In contrast to light field, bright field, where the complete it was hollow area. There was no central opaque. Okay. So here only peripheral rays will pass and fall on the specimen. The central rays will not pass. You can appreciate only peripheral rays are going from the hollow area. The central rays are going on the opaque area and they are not passing. In contrast to light microscope where all 4 rays were passing. Here only 2, the peripheral, the extreme rays are passing. You can appreciate in this diagram. 4 rays are coming, say yes. 1, 2, 3, 4. The extreme rays, the peripheral rays can pass because the peripheral area is hollow, but the central are not passing because this area is opaque. Central are not passing. Only 2 rays are passing. That's the point. That 2 rays will go through the specimen. They will reflect. They will go through the specimen. They will get reflect, and only reflected rays, okay, will pass here. From here the reflected rays will come. The reflected rays will pass from the objective lens, from the ocular lens. In light microscope, both reflected and unreflected were passing. Here only reflected rays are first passing from the objective, and then they are passing from the ocular, and finally that's why a bright image is formed on a dark background. Here the background is dark. Okay, so this is the difference. Only condenser is different. Now appreciate. This is light microscope. This is dark microscope. What is the difference in the principle? Appreciate. So here we have a light bulb. Here also we have a light bulb. Here also 4 rays are coming. 1, 2, 3, 4. Draw it. Here also 4 rays are coming. 1, 2, 3, 4. Draw it. Draw the condenser. People, draw the condenser. Here I have taken the same image. I'm sorry. By mistake I have taken the same image twice. Okay. I should have taken the light microscope image over here. Okay. I will take it. Give me a minute. Okay. Just a second. Give me a minute. I will take that image and compare it. By mistake, I have put the same image double. But no worries, I will put it from here. I will take this image and put it there. Give me a minute. I will share my screen again. You have to give me just 1 minute. Okay. I will share my screen again so that we can compare it well. Now, if I put the images back to back, you can easily compare it. I'm just sharing my screen. Let me replace that image. Now it's good. I guess you can see my screen. Yes, I guess you can see. I have replaced the image. Now see, it's correct. It was by mistake. You can see this is light microscope and you can see this one is dark microscope. Appreciate the light microscope, appreciate the dark. Now compare. You can see this is light source, 4 rays are coming. This is light source, 4 rays are coming. Now appreciate the condenser. Appreciate the condenser over here, which is completely hollow. Only periphery is there and nothing in the center. But appreciate the condenser. The periphery is hollow. The center is opaque. Appreciate the difference. Okay, so the difference is there. Here, all 4 rays are passing and they are going on the specimen, all 4. But here only 2 rays are passing, the peripheral rays. The central rays cannot pass. Okay, the 2 rays are going. So appreciate here, all 4 rays are falling on the specimen. Okay, 2 of them will be reflected, 2 will be unreflected. Here only 2 rays are passing, that is reflected. There is no unreflected. And after that, here reflected and reflected both are going in the objective lens and ocular lens. Here only reflected rays are going in the ocular and the objective lens. The unreflected rays are not there. Here reflected and reflected both are there. Here only reflected rays are there. And that's why here a bright image is formed on a dark background. Dark images formed on a bright background. Here bright images formed on a dark background. So that is a summary. This is how you have to learn, understand. So you can compare the principle of both. This is light microscope. This is dark microscope. Okay, please see my simplified flowchart. If you read them from book, it is, super confusing, but this is how you can simplify it. So here also we have a light source bulb. Here also we have a light source bulb. Here the light rays are coming and passing through condenser. Here the light rays are coming and passing through a special condenser. Here condenser is completely hollow. Here the periphery is hollow, but the center is opaque. So here all the rays are passing. Here only peripheral rays are passing. Here reflected and unreflected rays both are going to the objective lens and ocular lens. Here only reflected rays are going to objective lens and ocular lens. Ultimately magnified image is formed here, magnified image is formed here. But here dark image on bright background, here bright image on dark background. This is how you have to learn. I cannot simplify it more than this. I guess these are the heights. Yes. So what is the application of dark field microscope? It is used for thin bacteria. You know the thinnest bacteria are spirochete. They look like thread. You can see the thread-like bacteria. The thinnest bacteria, they are the spirochete. And they are used for staining living unstained cells. Living unstained cells. So these are the 2 applications. Here the application is the most common microscope. Here we have 2 applications. It is used for thin bacteria like spirochete and living unstained cells. Living unstained cell. You should draw the principle over here. You should draw the principle over here. Okay, introduction. This one form a dark image on a bright background. This one form a bright image on a dark background. So please see the introduction of both. See the principle of both. I have compared back to back the flowchart also, the diagram also. And see the uses, the application of both. Coming on the third microscope. The third microscope is phase contrast microscope. This is phase contrast microscope. As the name indicates, it increases the contrast of the image. Phase contrast, and there will be a phase difference in the wavelength. You have studied physics, you will be able to understand it better. So here the contrast is better, contrast is enhanced, and that's why it is known as phase contrast. You can appreciate the contrast is enhanced. I can't see only cell, but inside the cell I can appreciate this is the nucleus, this is the nucleoli, because it is having different contrast. I can see this is bacterial cell, this is nucleus, this is nucleoli, maybe this is mitochondria, like that. So different things have different contrast. In contrast to light microscope, where all the cells are seen similar. All the cells are seen similar. There is no contrast. Here there is no contrast, but here I can see the contrast. And that is the property of phase contrast. Here the contrast is better. Contrast is better. So what is the principle? You can see the principle, and I will show you everything over here in this diagram. You can appreciate here the light source is coming. The light is coming from the light source. You can see the light source passes through the specimen. So this is a bacterial cell from which the light source is passing. So this ray is passing from the bacteria, but this ray is not passing from the bacteria. So there are 2 types of waves, undeviated waves and deviated waves. Okay, the one which passes through the bacteria, you can see the green color bacteria. The one ray which is passing from the, both of them are coming from the bulb. Whenever they emit from the bulb, they have no phase difference. They are in the same phase. You know, the waves have a phase. You can see this is a wave. This is a wave. They are in the same phase. Okay, but this is a wave and this is a wave. You can see they are out of phase. You understand the phase, you understand the phase in the physics, huh? So whenever they are emitted from the bulb, they are in same phase. They are in same phase. Both the waves are in same phase. You can appreciate this wave and you can appreciate this wave. They are in same phase. Whenever they are coming out of the bulb, here bulb is not drawn. You can draw the bulb over here from which this is coming in the same wave, in the same phase. Now this one is passing from the bacteria and this one is not passing from the bacteria. So this one is known as deviated and this one is known as undeviated. Okay, deviated is out of phase by half wavelength. You can appreciate half wavelength. ये half wavelength is lagging, पिछड़ गई है half, because it is coming crossing the bacteria. This one is going in the air only, but this one is crossing the bacteria. Since the first one is going only in air, it is having its normal speed, normal phase. But the second one is passing through the bacteria. The second one is passing through the bacteria. You can see it is passing through the bacteria. So it is out of phase. It is out of phase by half wavelength. Okay, I'm sorry, not half, I'm sorry. It is out of phase by 1/4 wavelength. 1/4 wavelength ye pichhad gayi. So 1/4. It is out of phase by 1/4 wavelength. And you can appreciate here in the diagram. You can appreciate ye yahan par hai, but this is coming here. You can appreciate 1/4 wavelength ka difference hai. So the deviated one is retarded by 1/4 wavelength because of variation in mu inside the bacteria. Inside the bacteria, the speed will become slow, na? So this one is becoming slow by 1/4 wavelength. So when it's come out, both of them are out of phase. So this one is leading. ये आगे जा रही है. The undeviated one is leading. You can see this one is leading and this one is lagging. The deviated one is lagging. So undeviated one and deviated one. This one is leading. It is going ahead. This one is lagging by 1/4 wavelength. It is lagging behind. ये पिछड़ गई है. It is lagging behind by 1/4 wavelength. Both will reach the phase plate. This is a phase plate. Appreciate the phase plate. Can you appreciate the phase plate? In the phase plate, there is a hole at the center. From this hole, this one is passing, but this one is not passing because it is retarded by 1/4 wavelength. Deviated, undeviated, both will reach the phase ring. This one will strike the phase ring. This one will miss because it is retarded by 1/4. Okay, so this one will strike. It will pass through the phase ring from the hole of the phase ring. This one will not pass from the hole. It will pass from the opaque area. Okay, so this one missed the phase ring. And after that, both of them are retarded by half wavelength. यहाँ तक 1/4 wavelength से retard था. From here onwards, it is retarded by half wavelength. So this one is leading by half wavelength. This one is lagging by half wavelength. Okay, so both of them are retarded by half wavelength. Initially it was 1/4, now it is more 1/4. So total half wavelength. Total, when they come together, they cancel each other because they differ by half wavelength. No? So they cancel each other. When they come together, they cancel each other. They cancel each other, the deviated and undeviated. So undeviated one form the bright background and the deviated one form the dark image. So we get a dark image on a bright background like the light microscope. The only thing here, contrast is better because inside the bacteria, here there is nucleus, here there is nucleoli, mitochondria, whatever. Different structures have different mu. Please understand. Different. So nucleus have different mu, cytoplasm have different mu, endoplasmic reticulum have different mu, mitochondria have different mu. So the waves are passing from each point will have different speed and they all will have different speed and they will have different phases and they will form different brightness. So that will give the contrast. So within the specimen, different cells and different components have different mu. So wave will get retarded by different wavelength and that will give different light intensity and that will provide the contrast. I know it's very difficult. You can understand it, but how to put in words, putting them in words is difficult. So we will try. We will try how to write it simply. Okay, you don't have to go in the detail of the physics over here, people. You don't have to study the wave wala chapter in the physics. You have to simply understand so that you can pass your subject microbiology over here in university as well as an MCQ. This is phase contrast microscope. In phase contrast microscope, we have a bulb over here from which the waves are coming. Both the waves are in phase. They are in same phase. One wave is not passing through the bacteria. It is going by its normal speed. The second wave is passing through the bacteria. This is a bacteria. And inside the bacteria, different components of the bacteria, nucleus, cytoplasm, mitochondria. So suppose this wave is passing from the cytoplasm. Okay, so it will retard. It will reduce the speed. Okay, because it is passing from the bacteria, it will reduce its speed because here the mu is different. Here the mu is of air. Here the mu is of bacteria. Okay, so speed will retard. So it will retard from this wave by 1/4. This one is undeviated one. This one is deviated one. The undeviated one is fast. It is going ahead. It is leading. The deviated one is lagging behind. It's slow. It's lagging behind by 1/4. So this one will hit the strike plate. This one will miss the strike plate. ये fast चल रही है तो ये पकड़ लेगी hole को और ये छोड़ देगी. It will miss. Okay, when it will pass from the strike and it will not pass from the strike. So here the phase difference will be 1/2 instead of 1/4. Okay, and now they will cancel each out. They will cancel each out and they will form an image. Ultimately, a magnified image is formed. The undeviated one will form a bright background and the deviated one will form a dark image. So a dark image on a bright background with, like the first microscope, light microscope is formed. The only thing here, different components have different mu. Different components have different mu, and that's why the contrast of the final image will be different. Okay, you can see the images like this. You can see in the image, I can see the cytoplasm separately, nucleus separately, nucleoli, because they all have different mu. So when the waves are passing through them, the speed of the wave will be different and it will give different light intensity, that is different contrast. So this is the principle. Try to put them in the words and try to draw this diagram. That's it. Don't go in the detail, much detail. Okay, I tried to explain you. So here the difference is 1/4, here the difference is half. My flowchart is best flowchart. Just learn this flowchart by understanding. Don't just mug up. With the help of image, try to understand it and put them in words and finish it. Pack it. This is phase contrast microscope. Here there are two uses. It is used to see the motility of the bacteria. Can you see the bacteria are motile? We can see the motility and we can see the inner components well because the contrast is good. So for seeing the inner components, we use it. Coming on the fourth microscope. The fourth microscope is fluorescent microscope. Let's come on the fourth microscope. The fourth microscope is fluorescent microscope. You can see this one. This one is fluorescent. Here fluorescent colors are there. Okay. What is the principle over here? You know, here the specimen, that is our slide, our bacteria, we will stain it with fluorescent dye. We will stain the specimen before putting in the microscope. We will stain it with fluorescent dye. You will say, ma'am, what is a fluorescent dye? Fluorescent dye is it's a dye which is having a unique property. Whenever UV light pass on it, it converts the UV light into visible light. Okay, UV light is short wavelength. So basically a stain which convert the short wavelength into long wavelength. Long wavelength. That is known as fluorescent dye. So we will take our slide. Okay, this is my slide. This is my bacteria on the slide. I will stain it with fluorescent dye and then put under the microscope. And here the bulb is not simple bulb. Here the bulb is UV lamp. That is putting UV light. Short wavelength. UV light is short wavelength UV light. And when it falls on the object, it converts the UV light, short wavelength UV light into long wavelength visible light. You may have read this in physics, people. I'm just teaching you physics here. So short wavelength light is converted into long wavelength light. That is UV light converted into visible light, whatever you say. So say short wavelength converted into long wavelength or UV light converted into visible light. Such a dye is known as fluorescent dye. So before we put our slide in the specimen, we are staining it with fluorescent dye. So that is the introduction. You can see the principle in this diagram. I will show you in this diagram. So you can see here the light which is coming is mercury lamp. It's not a simple lamp. It's a mercury lamp which emit UV light, which is emitting UV light. From it, the UV light is coming out. Appreciate the UV light. It is coming out. After that, we are putting a filter. We are putting a filter that will allow to pass only short wavelength UV light. So only short wavelength UV light are going. Others are not going. Okay. After that, there is a mirror. There is a dichromatic mirror. Can you see? This is a dichromatic mirror from which these short wavelength UV light which got filtered, they are getting reflected. They are getting reflected and they are falling on the object. They are getting reflected. So from the dichromatic mirror, the short wavelength UV light get reflected and they fall on the specimen. The specimen is already stained with fluorescent dye. The specimen which is already stained with, before we put it in the specimen, in the stage, in the microscope, we are staining it with fluorescent dye. So as soon as we put the specimen over here, when the UV light is falling it, the fluorescent dye convert the UV into visible. You can see the blue color in the diagram is UV. Appreciate the green color is the visible. So short wavelength UV converted into long wavelength visible. Got it? So long wavelength visible is formed. So short UV convert into long visible. This is the main principle. And after that, it is going through objective lens. And after that, it is going through a barrier and it is going through eyepiece and a magnified image is formed. Objective lens, barrier, which will remove the UV and only visible will pass. And finally, it will pass through the ocular lens. And finally, a magnified fluorescent image is formed like this. A fluorescent image is formed. So what is the principle? You have to describe the principle over here. The main principle is, you can see in this diagram, the blue light converted into green. You can see. So the blue light falling on the object, you can see it is converted into green light by this object. You can see this is object which is stained with fluorescent dye. So fluorescent dye is converting the UV into visible, the short wavelength into long wavelength. That is the main principle. Okay, but you can describe the complete process. Say the complete process. In the complete process, we are having a mercury lamp over here. The mercury lamp is emitting the light. Okay, although it is emitting UV light, but we are still passing it with the filter, so that if any other light is there, it gets filtered, and only short wavelength UV light is passing from here. Now we will give a mirror. We will put a mirror over here that will reflect this short wavelength UV light on the specimen. This is our specimen which is already stained with fluorescent dye. So when the short wavelength UV light is falling on that, it is converting the short wavelength UV light into long wavelength visible light. So long wavelength visible light will come, that will pass through objective, okay, that will pass through a barrier, and that will pass through ocular lens. So first objective lens, then ocular lens, and a magnified fluorescent image is formed. I hope you got it. So this is how you have to write. Okay, I put much efforts to make these simplified flowcharts for you. You will not get these flowcharts anywhere in any book, people. So you have to write in the form of these flowcharts with this diagram. Okay, applications. We use fluorescent microscope to see plasmodium, okay, plasmodium parasite, malaria parasite, filarial parasite, TB bacteria, and immunofluorescence to detect antigen and antibody. You know what is immunofluorescence? I taught you in antigen antibody reactions in immunology. So that is the fluorescent microscope. The last, the fifth microscope. Learn the principle, people. The most important, I know it's difficult, but you have to draw the physics diagram. The physics diagram. You have to compare and contrast the first two. Okay, I have done. Okay, then the phase contrast, the two ways, 1/4, 1/2 wave retardation, and fluorescent microscope. And finally, I'm teaching you electron microscope. So let's come on the fifth one, the last one, electron microscope, the highest magnification. This one is having highest magnification of all microscope. That's why it is used for virus, because virus is the smallest organism, microorganism we know. Okay, so this is electron microscope. It looks like this. It's very costly. Very few electron microscopes are available in India. Okay, and it is used to see the microscope. The magnified image is seen on the screen, and you can see the magnification. The magnification is 1000 times better. You can see the structures very separately. You can see a magnified image 1000 times better as compared to light microscope. So electron microscope is discovered by a scientist. The name of the scientist is Ernst Ruska. Okay, E for electron microscope and E for Ernst Ruska. Okay, here we don't use light. Instead of light, we use electron. Electron. Why we use electron? Because the lambda, I mean wavelength of electron is less than visible light. Less than photon. You know, visible light contain photon. So we are not using photon. Till now, we were using a bulb that was emitting the light. This is a bulb. The bulb was emitting the light. Light mane kya? Visible light. Visible light mane kya? The light contain the photons. We don't use photon here. We use electron. Electron, because the wavelength of electron is less than photon. I guess you know this principle in physics. Huh? Have you forgot your physics? You can revise it here. So the wavelength of electron is less than that of photon, and wavelength is inversely proportional to resolution power. So since wavelength of electron is less, its resolution power is more. And this is how we are increasing the resolution power 1000 times. And that's why such a magnified image is formed. So instead of photon, we are using electron, because the wavelength of electron is less, and that's why its resolution power is more. Because there is a rule in physics, the resolution power is inversely proportional to wavelength. So the one who is having less wavelength will have more resolution power. The one which is having more wavelength will have less resolution power. Since electrons have less wavelength, they have more resolution power. You should be able to explain this in the simplified form as I have done. Okay, now tell me the principle. This is the principle. You have to draw this diagram. Step by step we go. So here we take electron gun. We don't take bulb. We take electron gun that generate electron. Electron gun is generating electron. We don't take a bulb that generate photon. We take electron gun that generate the electron. This is electron. So these electrons are passed through condenser. The condenser is magnet condenser. They travel in vacuum and they pass through condenser. These are the electrons that are passing through condenser. Can you see these are two magnets put? Okay, two magnets, and in the center there is vacuum from which these electrons are passing. This is magnet condenser. From the magnet condenser they are passing. Okay, we are not using simple condenser, hollow or opaque. We are using magnet condenser. After that, they go on the specimen. Here, the specimen is mounted on a copper slide, not glass slide. We take a copper slide. So this is our specimen. Can you see? This is our specimen, which is mounted on a copper slide. The specimen, the bacteria is present. So when the electron will go, they hit the copper slide. Okay, the electron will go. They hit the copper slide. The electrons get scattered. You can appreciate the electrons get scattered. The electrons get scattered. Okay, and two type of electrons get generated when they pass from the specimen. Okay, inside the specimen, the high density area, they scattered the electron more, and the low density area, they scattered the electron less based on the mu. So you can see this is the specimen. Inside the specimen, different components have different mu. So they keep on scattering the electron based on their density. The more dense area, they scatter the electron more. The less dense area, scatter the electron less. Like that. All the scattered electron go through the objective lens. They go through the ocular lens, and finally the magnified image is formed. They go through the objective lens, projector lens is ocular lens, and finally a magnified image is formed. So say the principle, try to say it. I know people, they are difficult. Okay, they are pure physics. Okay, but we cannot understand the entire physics inside microscope in just one session. Okay, people are doing PhDs in that. Okay, we have to see it superficially, we have to understand it broadly, we don't have to mug up, but you don't have to go in much detail also. Okay, you have to make a balance, you can understand, you can draw that. So we take an electron gun from which the electrons are generated. You can see these are the electrons. Then these electrons will pass through condenser. The condenser is magnet condenser. Appreciate the magnets. And in the center, they are condensing, they are going. After that, they fall on the specimen. This is my slide. The slide is copper slide on which the specimen, the bacteria is present. Now inside the bacteria, different components have different density. The areas which is high dense, they scatter the electron more. The area which are less dense, they scatter the electron less. And all the rays will fall on the objective lens, projector lens, and finally a magnified image is formed over the screen. So this is a principle. Revise it 2-3 times, try to see the diagram, try to draw the diagram, and you are done. Only one use. This is used for virus clinical specimen, tissue culture, immunoelectron microscopy. For viruses, we use electron microscope. So we are done. Now, can you tell me the differences between the first and the last? The first I taught you is light microscope. The last I taught you is electron microscope. You can see this is light microscope or bright field microscope. This is electron microscope. Say the differences. Say the differences in their principle. Here the magnification is 0.2 micrometer. Here resolution power. Here the resolution power is 0.5 nanometer. Don't see 0.2, 0.5. 0.2 and 0.5 is almost same. But see, this is micrometer and this is nanometer. Here the distance between 2 points should be in micrometer so that they are seen separately. Here the distance between 2 points should be in nanometer. Even they are seen separately. Got it? Here source of light is visible light. Here we use a bulb, photon. And here source of light is electron. We use electron gun. Here the medium is air. Here the medium is vacuum. Here we use a glass slide. Here we use a copper slide. Okay. Here the type of lens is glass lens, ocular lens, and objective lens. Here ocular and objective lens is electromagnet. Can you see? Both the lenses, ocular and objective lens, they are magnetic. Okay. Condenser is also magnetic and the lenses are also magnetic. So that's all. We are done with the principle of the 5. So you know the introduction of all 5. Say the definition of all 5. Say it. So light microscope form a dark image on bright background. Dark microscope form a bright image on dark background. Phase contrast, increase the contrast. Fluorescent, the principle is that if a fluorescent dye is there, if the specimen is stained with a fluorescent dye, so it converts UV to visible. It converts short wavelength to high wavelength. Okay, and here the source of light is electrons, accelerated electrons. Principle, please draw the 5 diagram. This is the diagram of light microscope. This is the diagram of dark microscope, dark field microscope. This is the diagram of phase contrast. This is the diagram of fluorescent microscope. And this is the diagram of electron microscope. See my 5 diagrams. I've explained you very well. Light microscope, dark microscope, phase contrast, fluorescent, and electron. Please see the 5 diagram, draw it here and tell the users you can learn it. That's all. I hope it's done. Thank you so much. If you have any doubt, you can ask it. Bye-bye.
Hello everyone. I hope you found this concept clear and easy to understand. If you would like to dive deeper into this topic and master it with full-length lectures, you can subscribe my app, that is MedLife by Dr. Priyanka. So join me for the live sessions where we break down the complex topics in a way that makes them simple and high yield for exams. See you inside.